So if you are even a little bit involved in science, scientific community, whatever, you've probably heard of CRISPR. If you don't really know what CRISPR is, it's basically a technology that is used for bioengineering and genetic editing and all that, and it uses the CRISPR locus and the Cas9 gene in order to make edits to specific DNA sequences to make specific proteins. But kind of what I haven't, at least on here, heard talked about quite often is the actual origin of CRISPR and what it's actually used for where it's found naturally in nature. Because, while it is a gene editing technology, it was originally discovered to be naturally occurring in bacteria. So the CRISPR locus was originally discovered in E. coli bacteria when some scientists were doing looking at genes and stuff, and they were like, hey, this is kind of a weird bit here, we should look a little bit more into that. And so they did. They found this CRISPR locus, and they found it in the E. coli and a couple other bacteria species and even some archaea species. But it was kind of unknown what the purpose of this was. They were just kind of like, neat, and then didn't really do anything with it for a while until some other scientists were like, hey, remember this, we should look at this a little bit more, and decided to dig a little bit deeper into the CRISPR locus and the Cas9 protein. So some more scientists kind of looked a little deeper into it, and it was discovered that the CRISPR stuff is used in bacterial immunity, specifically against a bunch of viruses. So kind of how CRISPR works is there's a piece of DNA and there's an enzyme, and the enzyme cuts that DNA at a specific sequence, so you can either change, add, or delete segments of that DNA. If you want to add a gene that makes a protein, you just stick it in there. If you want to knock out a specific protein, you just cut that gene sequence out, etc., etc., and then you get a new gene being expressed, and you get an entirely new protein made with an entirely new function. So you have changed the sequence of DNA to change the outcome of what that DNA codes for. So how the CRISPR system works in bacteria is it functions as the immune system. So it has similar functions but different mechanisms as to how our immune system works. So kind of what happens is there's a virus, specifically a bacteriophage, like these little orange guys. They will infect a bacterial cell and inject their genome into it in order to make new virus babies. And usually at the end of that life cycle of the virus, it kills the bacteria, and the bacteria doesn't really want that. So what the bacteria does instead is it uses its CRISPR system to cut up pieces of the viral genome and basically be like, this is mine now, and integrate it into its own genome for further infection. So it establishes memory, the same thing that your immune cells do after a viral or bacterial or whatever infection. So by using this little chunk of viral genome, the next time that virus tries to infect this bacterial cell, the bacteria is like, no, I remember you, we're not going to do that. So it basically acts as the memory part of the immune system. So the CRISPR system is very useful to both bacteria in staying healthy and evading viruses, and to us for gene editing and bioengineering and all that, which of course comes with ethical concerns and moral questions and stuff, but the technology itself is very interesting and has been used in the treatment of many diseases, as well as bioengineering and transgenic organisms, which I think is very interesting and people get very scared of for no reason. I've made videos on that before if you're curious. But yes, that's how kind of CRISPR works and the origins of it.